Hello friends, I am Ayantika and in this video I will be discussing about a very popular slime mold in your MSc syllabus, that is the Physarum polycephalum. Now the reason why the major scientists they are of much amazed by this Physarum polycephalum is the fact that despite this organism having just a plasmodial mass with no brain and no nervous system, have tremendous intelligence. Actually, in Hokkaido University, they experimented with Physarum polycephalum and they found that these organisms, that is the Physarum polycephalum, they were able to find the shortest route between two sources of food. And practically this shortest path problem, this has been used in biocomputing, and this is the reason why Physarum polycephalum is much studied. But apart from that, there are also several other experiments and analysis by which Physarum polycephalum has become much common in the lab analysis techniques. So today in this video, I will be discussing about the general aspect of Physarum polycephalum and its life cycle. So let's start the video. So Physarum polycephalum, just like the other slime mold, prefer to grow in cold, damp, shady places that is abundant in decaying vegetation, and they are cosmopolitan distribution, and they prefer mostly the tropical forest where it has got a lot of moisture and the forest floor is filled with decaying vegetable and leaves, and though it prefers tropical forest and it is found year round in the tropical forest, but in the temperate region also they are found especially during the rainy season. And few species are also found in the alpine region. Total, there are 100 species of Physarum, and Physarum cinerarium and Physarum compressum and Physarum viridae and bivalve. These are also common species of Physarum apart from Physarum polycephalum. So Physarum polycephalum has become most common because of the lab use. But apart from these four species of Physarum, they are very commonly found. So Physarum polycephalum, it is usually very large, but depending upon the nutrition condition, the size can vary. It can be microscopic to few centimeter to few meter, even few feet in diameter, but all depend upon the amount of food that is present. Generally, it is generally yellow, orange, brown or green in color, the plasmodium that I am talking about, and it feeds on dead decaying organisms as well as dead decaying matter as well as living organisms. That is, it is saprophytic as well as phagotrophic in nature. It is saprophytic, which means that when it is feeding on the dead organism or dead living matter, then it secretes enzyme, and by this process, if the food is broken down into carbohydrates or other simple form, and then it is just absorbed by the plasma membrane, or it moves by the it feeds by the pseudopodia. We know that the body is just amoeboid in structure for a considerable part, and so if there is a food matter in it. Then it by the pseudopodia, it absorbs that food, and that food absorbed in the food vacuoles, and then it is digested. It is the food matter is engulfed by the pseudopodia into the food vacuole, and it is digested. The food is digested in the food vacuole, just like that the amoeba does. Amoeba is another protist. It is an animal-like protist. So these are the two type of food feeding habits in Physarum slime mold. So the life cycle of this Physarum is divided into two phases. One is the somatic phase, another is the sporulative phase. So somatic phase, in the somatic phase, it is either two type, a very small amoeba-like structure called mixed amoeba, and another is the plasmodium. So both the amoeba as well as the plasmodium, they are acellular, which means that they, at this stage, this does not have a cell membrane, the cell wall. So amoeba and plasmodial stage, they do not have cell wall, but they are macroscopic, and they are multinucleated amoeba. The mixed amoeba, it is much smaller in size than that of the plasmodium, and the plasmodium can be formed by the fusion of many amoeba. But when this type of fusion occur between two or more amoeba, the nucleus, it does not fuse, and they maintain their individual identity. So amoeba contains a single type of nucleus, but it is the plasmodium may contain single type or may contain nucleus of two or more kind. But this whether it is amoeba or the plasmodium, they are diploid in nature, and as because they are diploid in nature, so this is the sporophytic phase, we can say. So this amoeba or the plasmodium, usually it is very small in the beginning, and then as it continues to creep over surface and feed on, they increase in their size. So when this plasmodium, it increases in size, and it is called a megaplasmodium, and after a certain period of gathering food, and if the condition is very conducive, then the plasmodium, it gets ready for reproduction. This is what we call that the plasmodium, when it is matured, it becomes ready for the reproduction. So this plasmodium, this is acellular, which means that they do not have a cell wall, but nevertheless they have the plasmodium and the amoeba, both they have a slime layer. It is very slimy. They have a slime layer. Then after that, the slime layer, they have a plasma membrane, a membranous structure, and then they have the cytoplasm, which is divided into ecto and endoplasm. And the ectoplasm, about the structure of the structure of the amoeba or the plasmodium, the cytoplasm is divided into two phase, that is the outer ectoplasm, which is much dense, and the inner endoplasm, that shows much streaming action. So this here we see that this is a plasmodium, where the anterior portion of the plasmodium, they are fan shaped, and the posterior portion, it has a network. So in the anterior portion, anterior portion has granular protoplasm, and the posterior portion there it has a network of tubes, and this streaming action that is found in the endoplasm is found here. So the network of channels that is present in the posterior region, they are showing streaming action. So whether it is this plasmodium, the ectoplasm, uh, the ectoplasm and the endoplasm, they have two different property, and this streaming of cytoplasm is observed in the endoplasm, that is in the inner portion. Say for example, the inner portion, they have, say for example, this is the ectoplasm. Ectoplasm is rigid and the endoplasm shows streaming. So this streaming, it was observed that this streaming is seen in a very cyclic event. That is, first it streams for, say for example, it streams clockwise in 40 to 60 seconds. Then after 40 to 60 seconds, the streaming stops, and then it starts streaming in the reverse direction. So the streaming is in a cyclic event, and this is called the cyclosis. So the endoplasmic streaming is called cyclosis, and this type of streaming is observed where? In the endoplasm, and also in the reticule-like structures that is present in the posterior region. So this region is granular. The anterior fan-shaped structure is granular. Not much of streaming is observed, but the posterior region has much streaming. Why I am discussing the streaming, the cyclosis? Because due to the streaming, information is sent as well as transport of food occurs. So the streaming is very much essential for transport of information, transport of nutrient, and even in the network in the posterior region, we observe that even nuclear organelles, they are transported by this streaming motion from one place to of the plasmodium to another. So also if any information regarding the external atmosphere, whether food is available or not, that is detected by this anterior region, and that information, it is transferred that information is transferred to the other parts of the plasmodium by this cyclosis motion. So this streaming is very much essential for the transport of information, transport of nutrients. So, and then back to the cell organelle. In the organelle in the amoeba or plasmodia, so they have an outer slime layer, and followed by the plasma membrane, and then the cytoplasm, which has ecto and endoplasm, and the endoplasm shows streaming, and the endoplasm, apart from showing streaming, it has got many nuclei and Golgi, titiosomes, and inclusions. Inclusions are food, they are stored food material, and apart from that, the most important thing is that the pseudopodial movement is caused by actin myosin. We know these actin and myosin fibrils, they are proteinaceous substances. Actin, myosin. So both this actin and myosin, which are ATP dependent protein, so they bring about contraction and expansion in the gel like portion of the ectoplasm, and the contraction and expansion brings about the movement of the pseudopodia, and also gives a pulsating force for the streaming action that is going on. So in this slide, we come to know about the somatic phase, that is the amoeba and the plasmodium, and the structure of plasmodium, about the streaming action, what is the function of cyclosis or the cytoplasmic streaming, and what the proteins, that is the actin and myosin, that causes contraction and relaxation, this pulsating wave of contraction and relaxation of the ectoplasm caused by actin myosin, which brings about the streaming, which is much essential for transport of information, transport of nutrient. So this is all about. And one thing is that this as it engulfs more food, it also keeps on increasing in size. So in case of amoeba, the nuclear division, it is open mitosis, that is nuclear membrane breaks down during the nuclear division, because there is no problem in the fusion of the nuclei. But in the plasmodial stage, as we have discussed, that maybe two or more type of amoeba, physiologically different, that they are not genetically similar, they have fused, but they maintain their individuality. So if two or more amoeba that has fused to form a large plasmodium, so they will have two different type of nucleus, and as because they have two different type of nucleus, so and they want to maintain their individuality, so when this two nucleus, when they divide, what they does is that they divide by closed mitosis, that is the nuclear membrane, it remains intact, and this is because mainly if it remains intact, then it is protected from fusion of other nuclei. So that is the reason why the plasmodium is having closed mitosis and the amoeba is having open mitosis. Amoeba is having open mitosis because it does not have to fear. So here, after the plasmodium has attained maturation by engulfing enough of the bacteria, protozoa, or fungi, so it has got enough food material, and it has got enough size. So after it reaches a maturity, then what it does is that it starts to reproduce if the conditions are perfect. But if the conditions are not perfect, then what will happen? That in the dark condition, or if there is certain drought or starvation or cold, then what happens is that the plasmodium, it converts to a structure called sclerotium. So there is two alternative pathways. One is that occurs in the dark and one in the light. In the dark or exposed to stress, plasmodium, this plasmodium is converted to a structure called sclerotium. Now sclerotium is just the plasmodium with thickened cell wall. So usually the plasmodium is acellular, that is it does not have a cell wall, but the sclerotia does have a cell wall, and the cell wall, it protects the sclerotia during drought or high temperature, and practically sclerotia remains dormant for a very long period of time, say for example 7 to 10 years, and only on return of the favorable condition do the sclerotia they germinate, and while it is subjected to stress condition, Sclerotia, it loses water, and it develops a cell wall, and it is divided into small units that are called macrocyst or spherules. So one sclerotia is converted into several small cellular structures called macrocyst or spherules. These macrocyst or spherules, they are covered by cell wall, and this each of the spherules or macrocyst in the sclerotia, if they get a proper condition, each of the spherules, they germinate into a diploid plasmodium, and each of the spherules, they contain one or more nuclei, diploid nuclei, and along with the organelles and plasma membrane and cell membrane, and germinate to form a diploid plasmodium. So this is the dark, and in the light phase what happens is that the plasmodium, it differentiates and form a sporangium. How it does it? So the plasmodium, when it is it is subjected to deficiency of food, or in a very dry condition, then what it does, it metamorphoses. The slime layer of the plasmodium, it dries up, and it stays as a dried the dried slime layer form a hypothalamus, and the entire protoplasm, that they form few mold like structure, few mound like structures, that is, for example, the plasmodium, this is the plasmodium. So entire plasmodium develops mound like structures, and eventually these mound like structures, they elongate, and gradually they elongate, and their tip becomes swollen. So this is how the sporangia is formed, and this sporangia, it is very brightly colored, just as the plasmodium was. We know that plasmodium is of Physarum. It is yellow, orange, with a reddish tinge or brownish tinge, and this this sporangia, which is formed by the meiosis, that is, this is a haploid structure. So what happens is that the slime layer dries up, and the plasmodium, it converts to a sporangia, and inside the sporangia occurs the meiosis, that is, from 2n to from the 2n to haploid, that is, from the diploid to haploid, the meiosis is done in the sporangia. So the sporangia, it is composed of this this structure is the sporangia, and this is the stalk, that is the sporophore. So this is the sporophore portion, this is the sporophore portion, and where meiosis occurs, it is the sporangia portion, and the sporangia, it is composed of an outer tough layer, non-cellular layer called the peridium, and the peridium, it protects the spores inside it. The spores, they are formed by meiosis, that is reduction division, where a diploid cell is converted to 4 haploid cells, and by repeated division, many spores can also be formed, and these spores, they are protected by inner structure, that is the capillum. Actually, when a single cell it is divided, it undergoes reduction division into four cells. The furrow, the cleavage furrow that is generated, it is filled with cellulose, and that forms the inner capillum. This cleavage furrow or inner, that is a capillum that is on the inner wall of the peridium, it protects the spores, and in fact not only protects the pores but also help in spore dispersal, because this capillum, they are made up of cellulose, and so we know that cellulose, they absorb water, and sometimes it loses also water. So by this absorption and loss of water, this hygroscopic movement, it causes bursting, it causes contraction and condensation, contraction and expansion of the capillum, that is and it causes the the wall, the peridium, the wall of the spore, that is the peridium, to burst and thus release the spores. So spores, they are once they are released by air, or if in a moist condition, by water, and also by many small invertebrate, especially the ants. So ants and mites, so they help in dispersal of the spores. So spore dispersal is of three types. One is by number one is by air, another by water, and another by. So this was what I was discussing in the previous slide, that the sporangia, they form the spores, and the spores, depending upon climatic condition, either remain dormant or they germinate to form the amoeba-like structure, or they develop into swarmers or flagellates. So the flagellates, they have an elongated body with anteriorly placed flagella. There are two flagella of unequal length, but they are both whiplash flagella. The longer flagella, it is anteriorly, it is directed forward direction, and the smaller flagella in the posterior direction. And the anterior region, it contains the nucleus. So the fusion of the amoeboid cells occurs of opposite strain. They also, the flagellates, they also fuse by this posterior end. So the two elongated flagellate, uh, they fuse by their posterior region, and they when the flagellates, they fuse, after the fusion, the zygote is remains quadriflagellate for a period of time. So after the fusion, this flagella, after a period of time, it is lost, and both the flagellated swarm cell zygote as well as zygote of the amoeba like structure, they have the similar structure, that is the fusion, the zygote, that is diploid in nature. So this zygote undergoes repeated division to form a plasmodium. This plasmodium, if after a period of getting eating, feeding, and maturing, if it suffers a stressful condition, then it is converted to dormant sclerotium, or there is also an alternative path, that is the reproduction. So this is the reproduction, and this is the dormant phase. Apart from that, there can be also haploid amoeba, that is this amoeba, when it does not find any any opposite gamete, then what it does is that these amoeba-like organisms, this amoeba-like structure, they by apogamy. So the Physarum, it has got three genes, MAT-A, MAT-B and MAT-C. We know that any gene has got many alleles, and recombination of these alleles brings about, increases the variations in the in the genes. So this variation in the genotypes as well as the phenotype is increased by alternation and of the alleles as well as the recombination of alleles of this three genes. And this is the reason why Physarum is used to study about the genetic inheritances. And this was all. You can go to through the, you can pause the video and go through it. I have been discussing this in the previous videos also about how the reproduction occurs. So this was all about Physarum polycephalum. I know that the video was long, but then I have discussed it in detail. And in any case, I have missed any point or you want me to make any new video regarding this Physarum polycephalum or any other topic, do write it in the comment section. Also comment on the video so that I can improve myself. And if you like this video, do share with your friends, and please do subscribe my video because your subscription will encourage me. And so far I have been making, I've started making videos on this fungi, and though the slime mold is not a fungi anymore, but I have started with this slime mold, and I'll be making videos on, the upcoming videos will be on various fungi. So thanks again for watching this video.